really just to set the stage, there is a lot to cover and of course a lot of complex topics to cover, and I will do my very best to make it clear, but please do feel free at the end, probably easier at the end, otherwise we may not get the material, to ask any of the questions either related to what I present or if I haven't mentioned something that you might be interested in. And I thought I would focus on 3 specific areas. First of all, look at ALS biology. For many of you, the disease is extremely familiar. However, it's really through understanding the biology that we can make progress. And although in some ways we are doing things in parallel, we don't have all the answers from biology, and we are launching forward with treatments, it is good for you to be able to visualize what the disease really is about. I will talk briefly about our research programs and our goals, and then talk about current advances. I may not remember to mention that most of the current advances I will talk about in some way has been funded through our support and through your collective commitments to help keep the ALS research program going. So I won't say this is Elsa's study and this is, but essentially we really fund in some part most of the work that is going on globally. So it's very exciting to play a big role in this, whether we fund a small amount of a particular study or it's a partnership. We are able to impact work, and it's global because we don't know where the answer will come from. So this I like to show as a visualization, and we talked a lot before the evening about the fact that motor neuron disease, most of motor neuron disease is ALS, and ALS is when the upper and the lower motor neurons are involved. What does that mean? Well, the upper motor neurons are in the motor cortex in the brain, the lower motor neurons are in the spinal cord, and all of them are exquisitely connected so that function can happen normally with these very long processes which we call the axon. So visually looking at it, there's a very complex architecture that leads to finally the miscommunication of information and the inability of the muscles to move. I show this image because there'll be many of you that might say, well, I've listened to a whole evening of Lucie and I still haven't heard about the cure and about a phenomenal treatment. I don't promise you that tonight. One thing you have to realize that this disease is an incredibly complex disease, and so we have to start to understand these connections to try and recognize how we might treat it. We'll talk a bit more again about stem cell therapy. If we think about Alzheimer's disease, another disease actually occurs very frequently in aging populations, very difficult disease, billions invested by the pharmaceutical sector, and no treatments either for that disease. And they have the fortune, if there's any fortune involved in it, to have what we call a focal disease. One region in the brain is affected. So remember, when we think about ALS, it's a complexity of cells and all their connections that we have to think about, and we have to think about repairing. So that's quite a challenge. So most of the years prior to 1993, what we thought of ALS was that it was a motor neuron disease. The key thing is the motor neurons. These are nerve cells in the brain and the spinal cord. And they have, as I mentioned to you, a very small cell body with a central nucleus and a very long projection. This is a normal cell in a human. This is one where there is ALS and the cell is dying. And we saw these very abnormal swellings and these accumulations. So prior to 1993, when no mutations had been identified, and we'll talk a lot about genetics, this is what we saw of the disease. Abnormal neurons, clumps and things accumulating, and very little else to go by. And of course, many of you know that this was discovered by the Frenchman Jean Charcot in the 19th century. So it's been around a very long time and been described, and not much had shifted. And these are images prior to 1993. So what has happened in the last decade, I would say, is a complete change in how we think about the disease and the complexity of the disease. So not only is the motor neuron, and we always show images of motor neurons, it's actually a motor neuron, it happens to be a mouse motor neuron. It has that small cell body. It does have one single axon, but it also has a complex network of other what we call dendrites. So the picture is not as simple as saying a cell connects by a long string and then connects. It actually has a network of other things that have to interconnect in the proper way. So it's a very complex cell. Next to it and surrounding it, we have what we call the astrocytes. These are stellate, much more decorative cells that are also very important. What do astrocytes do? Well, they surround and they actually make physical junctions with the motor neuron, and they are critical to keep the surrounding of the motor neuron in good health. So astrocytes, and this has been described by Jeff Rothstein and many others, but Jeff Rothstein was one of the key players. He has showed that these astrocytes suck up and mop up glutamate. Many of you who have heard of glutamate, often concerned glutamate might be toxic. Glutamate is a very important transmitter in the brain. You have to have glutamate to send the correct messages. Too much glutamate, though, is indeed a problem. And when we talk about glutamate toxicity, it's because there's an abnormal amount of glutamate surrounding the neurons. These astrocytes are there to mop up and regulate, and we do know that in ALS, there's something wrong with the astrocyte in being able to regulate that glutamate. So a very important cell. Then I show this image, which I think is spectacular. What you can't see, which you might see a little bit here, but what is in black, is actually the muscle. And so again, it's not as simple as a cell body with a long axon attaching to the muscle. There's a huge network or arborization, looks like a Christmas tree, of connections that will touch the muscle. All of that has to function properly. Those little things we call boutons, or buttons really, so to speak, are the chemical connections. So you've got your messages gone from the neuron all the way down. It transmits chemically to cause a muscle contraction. And we know, this is actually in the mouse model, we know that that arborization, that connection with the muscle, is completely destroyed if we manipulate the mouse to have ALS, and I'll show you how that is done. So we have to worry about the surrounding cells, the motor neuron, the connection with the muscle. Much more complex than just the motor neurons. And so when we think about ALS, and everyone would love to say and love to know, what was the cause? What was it in my case? What were the first things I saw? Was it perhaps that I'd had some injury and therefore I got the disease? The reality is that all these components come into play. Aging, it's most typically a disease as you age. It's not certainly in very young, although there are exceptions. It's generally speaking only 5 to 10% genetic cause, where it goes directly from family to family. That said, genes play a key role even in sporadic cases of the disease, and there are a few studies I'll mention about that. And then it's a combination of the environment. We know about the military, the twofold increase. Why is that? We're still trying to work that out. Is it the smoking? Yes, we know that that is an increased risk. Is it the pesticides? Golf players exposed to pesticides on golf courses. Many theories. More recently, NFL players affected by trauma. Soccer players in Italy. Marathon runners, a study done in Ireland. So physical activity, Lou Gehrig himself, maybe, all very difficult to measure, and my gut feel is it's a combination of factors. It's the susceptibility that you have, why someone else who runs marathons doesn't get it and you do, is because of a genetic susceptibility. So a little bit about our programs then, and as Becky mentioned, one of our key goals, it's great to fund good research, it's fantastic to do some interesting studies, but my passion, I'm a pharmacologist by training, that was my first degree. I then went into specialize actually in Alzheimer's disease, and after working in ALS and building one of the mouse models I'll show you, I went into the industry because actually my passion is developing treatments. That's the whole goal of understanding biology, is to actually be able to find treatments. And so although our program initially had some very good project ideas, I helped the organization move this from just being good labs, doing solid research, and helping see how we can move it. The industry likes to talk about a pipeline from understanding and getting some early clues all the way down to developing drugs. It's a very systematic process in terms of developing drugs and testing them and then getting them into clinical trials. Most important to actually achieve all of this is to build model systems, we'll talk a little bit about that, and to have biomarkers. So there is one key example, I won't spend too much time on it, where this really did happen, because sometimes it's nice to see these schematics and think, well, it sounds all very good, but does it actually happen? Well, a very quick example, and it's hard to put it in the logical order, that I'll tell you more about SOD in a minute, but this was the first genetic clue in 1993. It happened to coincide with the time that I got into ALS research, and it moved the disease from just looking at blobs and motor neurons to trying to tease out disease mechanisms. So it was the first big genetic clue for familial ALS, patients that have it tracking in families. And with this finding in 1993, I worked with Don Cleveland, one of the key ALS investigators, to build models to question what is SOD1 doing, and we'll talk more about that. We have still discussed what is SOD1 doing, but a few scientists in Don's lab, including a clinician, thought, what if we can just get rid of the bad protein? Don't worry about what it's doing, we know it shouldn't be there. Can we get rid of it? And so a project came through into our programs in the early phase, very risky, a crazy idea, uses technology that a company, ISIS, has, never used in the neurological disorders, only for others cases, but never for diseases like Alzheimer's and Parkinson's, and thought, well, let's give this a try. Essentially, they blocked the production of this protein that we don't want, and it moved from being an idea all the way into the preclinical studies, very expensive. We had to reconfigure our programs to fund the several million it took to actually fund the preclinical studies, and it's gone into clinical trials. It's completed the first round, it's safe, and it's in a process where, because of so much that has been learned, they are redesigning it slightly to then go back into the clinic and to have an improved effect. But this is for the 2% of ALS, I know a small amount, that carry the SOD1 gene. But that technology has opened the doors up for Huntington's, for SMA, for myotonic dystrophy, and now for the new gene I'll tell you about later. So a simple idea that came from the laboratory, we were able to get it into clinical trials, and it would not have happened if it hadn't been for our research programs, our ability to say we will fund the biotech company, because actually they don't focus on ALS. Sometimes it's odd to say, well, why are you funding a company, a for-profit organization? But we incentivized them and made them realize that it wasn't such a risk to take on board ALS. And now we don't have to provide any funds anymore. They've got more than sufficient to take it through to a treatment. So this is maybe a busy slide, but it's easier than having written things. And this is the translational program. And it has many different approaches to it. It's not so important to remember the terminology, but there are many different ways we are able to support and provide funding. The investigator-initiated awards program is when people send their ideas to us, because a lot of work and many organizations now are doing much more of a top-down, we're going to dictate what should be funded. That's a good approach, and we do that too. But sometimes ideas come from crazy places, and we see all kinds of things coming through this open solicitation. Bring your idea, we'll try and see which of those we can select for funding. We then do the top-down approach. This is sort of more bottom-up, the top-down, where we identify with my scientific colleagues and advisors and some great leaders in the field, Tom Maniatis, who's been highly recognized for his contribution worldwide and now focuses on ALS, many Nobel laureates who are advisors to us. And we identify where the gaps are, and there have been few major breakthroughs through that recognition and that kind of program. Particularly good to build consortium teams, and we'll talk about that later. We had years ago, and we have just relaunched our drug discovery contracts, because I think it is through thinking about how we develop treatments now, even though we don't have all the answers of the biology in the early part, we need to do these things in parallel. We can't invest in worrying about all the details of how the disease happens without a parallel trying to take the risk and build some therapeutic approaches. So we do it at the same time. We also fund small pilot clinical trials like the antisense study I spoke about. We have a network of clinical trials and clinical centers that we help to quickly mobilize clinical trials. And we fund clinical management awards, and I'll speak about a few of those. All of them are interconnected. The teams of people are often interconnected. So we feel that there are many ways to approach, and it's very important to approach many different types of schemes to fund research. And this is just to give a numbers sense. And one of the key things as well, do we have enough projects to fund with a pool of money that comes in? And in reality, we have a lot more than that we can actually fund. For one particular program that I launched this year, the drug discovery contracts, I anticipated about 10 solicitations. These were big awards for a total of $1 million. And we got actually 60 applications, so we could only select a few of them. This is giving you a sense of where we put our money and how we try and spread it. The drug discovery area is the most expensive, together with clinical trials. So we have about, in our portfolio, the value of $10 million worth of research. Might sound big to some, very small to others. Is not meant to make any other statement than to say a significant amount is invested, but we need much more to be able to get where we need to get to. The other thing that we do that I think is an important part of it, though, is that small dollars can go a long way if you make successful collaborations. And that example I showed you right in the beginning with ISIS, the company, where we invested a couple of million and they've now put in 23 million. We could say, well, we spent 23 million on our program because we managed to mobilize that kind of activity. So numbers are not always reflective of success, but it takes a lot to actually get these research programs underway. So then to go into, and I'm happy to talk about any of these in more detail at the end, but to give you some of the updates in terms of what is currently happening. And genetics, I have a little bias, I'm a molecular biologist, and genetics is much more tangible than doing environmental studies. How many people are exposed to a certain thing in the environment and what are the key toxins? Very challenging, not unimportant, and we have epidemiologists who are funded to do that. But with genetics, you can find an error, you can make that manipulation, and you can see what goes wrong. It's much more easy to do. So the most remarkable thing is the last few years in genetics. When in 1993 I joined the ALS field, we were talking about this piece of the pie. That's 2% of all of familial, remember this is familial ALS. Familial ALS is when there are more than 2 cases of ALS in a family. This becomes a tricky area when we start to talk about some of the other genes, which are appearing in sporadic cases. Where we see genes that are strongly involved in familial ALS, also in sporadic disease, the question does become, do we really know the family well enough? And are we missing members of the family who have had the disease? So it is a little bit of a tricky area, but familial ALS is generally if there are more than 2 cases in a family, 2 or more cases in a family. We only knew about 20% of familial ALS, and that's SOD1. After that, in 2006, we identified 2 other very interesting genes, FUS and TDP43. I won't have much time to talk about them, but they are about 4% of familial ALS each. So this year, and we'll talk a lot more about that, the major finding, which has almost completed this picture, at least more than half, is the C9orf72, a change, a genetic change that links a lot of familial cases, in some countries 40% of familial cases, and also some sporadic cases, and we can talk more about that. So this is extremely exciting, and my colleagues who tend to not overblow something, they have told me that probably within the next 5 years, if not sooner, we'll have solved all the genes for familial ALS. That's due to technology, that's due to increased commitment, and finally finding C9, and I'll tell you a little bit more of a story about that later. But what does that all mean for you? I mean, it's great. So we've got a big piece of the pie is filled, and we're all excited as geneticists. We have all these genes. Well, the genes have given us the clues to start understanding disease, and I'm sorry if I'm blocking you here on the side. So this is just an image of superoxide dismutase, SOD1, superoxide dismutase 1. When in 1993 this was identified, we suddenly had a great number of biologists very excited because They'd known about SOD1 for years. Superoxide dismutase, as some would like to say, was a protein looking for a disease. Because they knew all about this, they could solve the problem once they'd found mutations. I wish it had been that simple. What we see in this image here, so mutations are basically, if you have, so to get a protein, you have to have your DNA, which is your messaging in your nucleus. From there, you have to then have a matching strand of RNA, and some of you might remember this from your biology classes, and then the RNA will turn magically into protein. If there in your DNA is an error in one of the codings or the messages, that is taken down as an error into the protein. In SOD1, there are over 120 errors. It can be at different places, and that's why A4V, which might be familiar to some of you, A4V SOD1 is the most common SOD1 mutation in North America. Actually, because of a common ancestry that came over to North America, that's why it's so prominent. These are all mutations that cause a change in the protein. So normally, what does SOD1 do? It's in every single one of our cells. It's in our lungs, our liver, our spleen, our brain, our spinal cord. This is the most remarkable thing about it too. It's everywhere, and it's in a cell to make sure it gets rid of toxic waste. So it's basically regulating increased oxidation that happens from normal cell processing. That's its normal functioning. However, when you have a mutation in SOD1, and those families, I don't know if there are families here, but where they have a mutation in SOD1, they have one normal functioning allele, we call it, one that still functions and one that doesn't. And we recognize that the function, the normal function, is still working in patients with ALS. So we're getting rid of the toxicities. It's doing something else. But having learned about these mutations, and this was my field of work when I went into ALS, we were able to start building model systems. And obviously the best test to understand something is the human itself, but it's much harder to look at windows in time. So what we were first able to do is to try and manipulate cells in culture. But what was more exciting, and this again is technology, we were able to manipulate the genetics of a mouse and introduce into it the human mutation that is linked to ALS. And again, what was remarkable to me, and this was my first experience of building a model like this, is that this particular protein is, as I say, in the lung, in the liver, in the spleen, everywhere in the body. And we put the mutation in everywhere in the body. That's how it works in the human case. But you get ALS, a motor neuron disease, also in the mouse. So at midlife, at their midlife, they start to have a hind limb that is weak and drags it, and then the other one spreads to the other one. And so it looks in many ways like the human disease. So we can now, in a model system, manipulate it. Since then, we've worked from building mouse models to rat models, worm models, fish models, fly models, anything that we can manipulate. And you might wonder why all these different things. Well, each of these systems has an advantage and an ability for us to test the biology. A fly, for example, has a very short breeding cycle, so you can analyze things very quickly. You can actually test drugs on them. Same with worms. And in fact, the worm model where we put the mutation in SOD1, what happens to the worm is it doesn't move as fast anymore. And so when you start to treat it with drugs and you ask, well, which kind of thing, if you give it to that worm, will help it move better? And you can test this in a much, obviously, quicker paradigm than testing every single one of those, either in the rat model or, of course, in humans. So they are fabulous model systems to test concepts and to understand disease. Another very recent finding was that the fish model was a very good tool. And what can be done in the fish is that you can actually, in live time, see where the connections of the neurons are, because it's see-through, and you have special marking tools so that you can now see these are the motor neurons. And what this group did was look at the motor neuron projections. Remember, I said to you that there's a very long process that connects to the muscles. Well, these are those in the fish. Here there's the cell body, these are the long connections, and then they connect with the muscle. And we find when we put the mutations in that these connections are abnormal. And in fact, in a very recent study, and you might have seen it on the update in the news, this same system was used and a variety of different drugs were used and identified that there's a specific pathway involved, at least in these fish models, and that same abnormality is found in some human cases. So the worm and the fish might seem a long way from the human, but when we go back to humans and test our ideas, we often see that they actually correlate. So these are fantastic tools to work with. It does, however, make ALS a much more complex picture than we could ever have imagined. We know that there are many cells involved. We know that there is a connection between the cell and the motor neuron that seems to be, and the muscle that seems to go wrong. And all of these components are areas that we fund, areas that we study, and we have to worry about, because it's not only the astrocytes that mop up the glutamate, it's the cells that are involved in inflammation. And in the brain we have special inflammation cells called microglia, behave differently to inflammation that happens when you cut yourself and you see a swelling. But as such, there's also an injury response in the brain and spinal cord. Some of it is good and some of it is bad. So with this picture, it might be a morass to us to say, well, where do we start? The reality is each one of those are what we call targets for a treatment. Approaches that we can now think about to develop a whole variety of drugs. There might already be drugs available that we can test according to these things. And it is why I think it's going to be very difficult to imagine that there will be one treatment that does it all. It's probably going to be a variety of approaches to tackle the disease and cause a big effect on the change in survival. Some examples, and I'll talk about clinical trials, but some examples, the key target is the mitochondria. Dexpramipexole, the Empower study at Biogen, we think is because we are improving energy metabolism in the mitochondria. And there are many different other approaches. We'll talk about a compound that is being used to improve muscle ability, to use your muscles. So there are many ways to think about therapies, and it's because of understanding the biology and because of these mouse models where we can look early in time, as soon as things are happening or before things are happening, to try and understand what the cascade of events are. Because one of the huge challenges in treating this disease is that we actually are identifying ALS too late. In many cases, the disease has progressed so far that, yes, we might be able to stabilize, and that's absolutely our intention, but you're not going to get back what you have already lost. So what we really need to do is find earlier treatments and be able to diagnose earlier. Another clue that came a large part from genetics and also from a finding by Virginia Lee's group is the reality that ALS is not only not motor neuron disease surrounding cells, it's also affecting a type of dementia, not Alzheimer's disease, but it's called frontal temporal dementia. Not in every case of ALS, but in many cases of ALS, there's a cognitive change which is different to Alzheimer's disease. It's actually more a behavioral change than a dementia in Alzheimer's. And what we are finding is that many of the genes that we have identified in ALS are the same genes that we are seeing in frontal temporal dementia. This is important to know because, in fact, to help and look after a person who has frontal temporal dementia is a little bit different to someone who only has ALS. And we think, in fact, clinicians really now define a spectrum. You can be on the one side have a pure ALS disease, on the other side a pure frontal temporal dementia, and then in between a combination of both. And so sometimes at this point, if you only have ALS, later on you will progress to get ALS and FTD. In some cases you will never progress to have FTD. But this is an important thing in understanding how to manage ALS, and it's a reality that many are not aware of. So the identification of this particular, and it's an awful name, and we hope that it will eventually be named a better name, but the reality is nobody knows the protein function of this is not known. So what happens is, I'll just tell you exactly what it means. C9 is chromosome 9, a region on your genetic coding where this mutation is found. It's in an open reading frame, so it's an area that does not translate into a protein, so it's not transcribed into a protein, and it's at position 72. Until we know what this protein is, we won't be able to make up a very good name for it. We don't know what it does, we don't really know anything about it, other than that it is in the spinal cord and the brain as well as other tissues. It was the first genetic clue that told us that FTD and ALS had something strongly in common, because this gene is the most dominant gene mutation found in frontotemporal dementia, and it's very prevalent in familial cases. It was identified first in a family from Finland, where 6% of the families are now linked to C9. So a very large proportion of ALS is linked to this. And in that particular group, 21% are in sporadic cases. We're not sure actually if those are truly sporadic cases. So what are we trying to do at the moment? So C9, and I'll show you a little bit of the biology, was very unusual, and actually the story is well worth telling. We funded 3 different groups to identify this. We knew that there was a link on chromosome 9. It was also extremely interesting families, because in some cases they had, one generation had frontotemporal dementia, the next generation had ALS. So it looked as if it skipped generations, but actually it was either frontotemporal dementia or ALS. We knew these families, and we've been working on it for 8 years. We at the association had put together a consortium of the best scientists, the top people from the Human Genome Project. We put several million into the program, and we were convinced that we would find these mutations. It was a very small region, and to tell you the whole process of genetic finding is difficult, but it's a very small area, very few actual genes in that, so it should have been easy. And the team didn't find it. At the same time, we funded 2 small teams, one at NIH and one in Florida. Young, it's up-and-coming Brian Trainer and Rosa Redemakers, 2 very good clinical scientists, but early in their career. And it so happened that those were the 2 teams that found the mutation. And not because they did it through any fancy technology. In fact, we funded the best technology we thought to do this, but using old technology. Because in fact, the new technology was constantly throwing out this data as if it was an error. So it's not always the biggest, the best, and the most fancy technology that does it. And we have to fund sometimes many studies looking for the same solution. And the fascinating thing is, we didn't know, didn't think that ALS would be caused by repeat disorders. Now, Huntington's disease has these enormous repeat expansions. There are many other diseases that have that. ALS has always had one single change in the genetics to cause the disease. In C9orf72, there is a region here on this gene, and you don't have to absorb any of that detail, but just to say, here in this area here, normally this is the pattern you should be seeing, and you should have about 20 of those repeated. In this case where there's a mutation, you have at least 1,000 of these, an enormous chunk of repetition. What new technology does is it splits it up nicely into small packages, and you think you've only got one of them. It didn't identify that you had them all stuck together as one string. So this is the problem. Do we understand it? Not really yet, but what we can speculate, and what we've seen, and these are patients that carry the C9, is that we get these unusual blobs or clumps. So in other words, having such a big chunk of stuff you don't want to have there, the cell is unable to deal with it, and it starts to link together and make a big blob. What is very exciting about this is that I told you right at the beginning, with the SOD mutation, we spent years trying to figure out what it does, and we intend to continue to do that, but we thought just get rid of it. Well, now scientists, while they are thinking what this protein does and where it goes and what's happening, are already going straight to the point and saying, well, we know it's not supposed to be there, so why don't we just get rid of it? And so the same company that worked with antisense technologies for SOD1 is doing the same approach, and many other companies now, because they see it's a very lucrative area to be in, are looking at blocking that production. The exciting part of it is it's not going to take 13 years of figuring out what's going wrong and then another 20 years of drug development. We are working already now to start thinking about how to treat people that carry this mutation. and it's in frontotemporal dementia, it's an ALS, and it's in a large proportion. So I think it would be an incredible and very, very possible opportunity that we can treat a significant part and maybe cure a significant part of ALS. Early days still, but the tools and the technology are being built towards it. So that's enough genetics, but moving now along to stem cells. And I'm only going to briefly talk about it because I didn't actually check to see how much time I have, but I will keep going until everyone's asleep. No. With stem cells, so many of you have heard about this, and I have one question here as well about stem cells that I got in advance. The question about why is it that bulbar patients are not accepted into stem cell trials? Well, first of all, there's only one stem cell transplant trial going on in the US at the moment. That's a neural stem. And the trial was set up because there's such concern from the FDA for safety, which is reasonable because this has not been done in ALS and any disease successfully other than cancers and replacing that kind of stem cell. That they had to, at that point, take patients that were not mobile, and many bulbar patients are very mobile. That's the one reason I can think of. But there is no reason to say that we will only treat one sector of bulbar versus limb onset patients with stem cells. But we are quite some way from being successful in using stem cell transplants. I'll just show you this image. It's an incredibly invasive technology. So the stem cells are injected into the spinal cord. The spinal cord has to be opened. This is all done by work funded by the association. Nick Boulis is the neurosurgeon for the neural stem trial. And what he does is he first injected obviously into model systems before he went to humans, but he adapted this in such an elegant way that he knows exactly where the cells are injected into the spinal cord, and they basically do 5 injections on each side of the spinal cord at one time. It used to be only in the lumbar region, but now the FDA has approved, because of the safety concerns that have no longer been seen, in the cranial region. That was much more sensitive because there could be much more damage done there. And so far, everything has gone quite well. People have not died because of the procedure. It's been quite safe. They do have to be on immunosuppressants, and most of the people in the trial have had more trouble with the immunosuppression than the actual surgery. But there's no indication, it's not expected, that there's any dramatic effect in terms of extending lifespan. This was just to test safety. One would have to treat probably much earlier in the disease. These were very late patients for safety concerns. But that's transplantation studies, and a lot of work is being done, and there are 3 major groups that are planning towards clinical trials in the US in the next year or 2. So there will be follow-up studies. This set the stage and actually got over the first very difficult hurdle of doing such an invasive treatment and such a risky treatment. There are stem cell clinics all over the world. If anyone wants to talk about it and wants more information, I always encourage you to talk to your neurologist. You're very welcome to talk to me. I would be concerned if there isn't any rigorous follow-up and there's been some crazy things going out there, and no real place to say that they actually are helpful. They might say that on their website, but there are no success stories that I'm really aware of. But stem cells have had another explosive and exciting expansion and an opportunity. And this is IPS, induced pluripotent stem cells. And this finding has been phenomenal. One, it's got rid of all the ethical concerns. So I still get people and chapters and donors emailing me and saying, well, we won't fund your organization because you support stem cell research. Actually, in our portfolio, we don't fund any embryonic stem cell research at this point in time, because induced pluripotent stem cells are such an opportunity that we've invested more in that. This is a team of well-known clinicians, biologists, and scientists, both in industry and academia, and we support a partnership at the National Institutes of Neurological Diseases and Stroke, NINDS, to build up a resource of these cells for people to use worldwide. Now, what are they? So basically what you can take is a skin cell from a patient, and the good thing about this is You don't have to know about the genetics of that patient, especially if they're sporadic. They wouldn't be an SOD1, they might not be a C9, but we are capturing their genetics by taking their skin cell, taking them into a dish, manipulating them with some particular genetic coding, and then moving them back into embryonic state so that they can develop motor neurons. So it's quite phenomenal. From a skin cell, you can generate all the cells of the brain that we're interested in for ALS. And carrying with them are then the errors, the genetic errors that are associated with the disease. And so this becomes an incredibly rich tool for us to work with. And as I mentioned to you that the new C9 mutation has been identified, we now, from this large repository and this large resource, have scientists who have been able to take all those cell lines and started to look at very specific experiments to see how long that repeat is, all kinds of very, very clever things. So we didn't even know that that would be the value of them because we didn't know that that genetic discovery was coming along. Another thing that Steve Finkbeiner has done, an investigator we fund, is to work with these cells and set up a microscope in which it's all automated. He can follow them over time. So it's very impossible to really make a human model in a dish, but we do our best to make it as real as possible. And so what we do is we have this cell that now is the motor neuron, and we follow it over time. And we say what goes wrong and how can we change it, because this is all set up as well to add drugs to those chambers to see if you can change something. And then you can film them and measure them over time. And through this technology, he has been able to, and has published, and we've put out a news release on this, identify one of the mutations, the TDP43, I didn't have time to talk about it. That mutation was harbored in the skin cell and therefore in the motor neuron, and he could show the abnormalities that were happening. There was a strange accumulation, and he is now using that to test which kind of drugs might reverse that. So very real opportunities to do drug discovery. And this is just showing you how many lines and the different lines we've now collected. So that was a very quick tour of stem cells, only to say transplant studies are occurring. There is a lot of work being done, much more challenging, but we have shifted to using induced pluripotent stem cells as a possible tool for drug discovery. So biomarkers is I could spend a whole evening talking about biomarkers, but I did want to recognize that this chapter has been phenomenal in helping move that forward through the specific fund that was set up. And those funds have gone to very, very important studies, and biomarkers are key. What is a biomarker? A biomarker can be a measurement of muscle strength, it can be a blood test, it can be an image of the brain. It's any indication that someone has the disease. And more importantly, if it indicates that the person has the disease, how does that look over time? What happens during the progression? If we had such a solid biomarker that changed over time, and we could then use a drug and say, when we treat, that marker, does it that image we had, does it change over time? Does it improve over time? That is actually, I believe, why multiple sclerosis has so many drugs on the market now, because they made a breakthrough in their discovery of finding a biomarker for the disease. And companies now have invested heavily and have found treatments in a much shorter period of time because they would look at the change of that image in time. What happens in ALS clinical trials at the moment, and some of you are aware of it if you are in trials, is that yes, you do functional measures, the functional ALS-FRS, the functional rating scale, largely measures of quality of life, how long is it before you can't do something anymore, the decline in function. And the main endpoint is survival, which can be 2 to 3 years in some cases of the trials, and other cases much longer. So that's an incredibly difficult measure if you're trying to test in a company whether a drug is working. And so until we change that, and certainly the Biogen trial is carrying on with the ALS-FRS score and survival, and they've done an interesting ratio between the two, but until we really get a good biomarker, many, many companies won't significantly invest in ALS because it's a challenge. It's too long, it's an unknown end. So that's a critical area, And what are we doing about it? It's again not easy, nothing has been so straightforward, but we are able to image the brain, and we are able to see some cues, either chemically or by physical structure, that is different in a person with ALS versus a person who doesn't have ALS. Our challenge at the moment, and we hope with better imaging, we're not seeing significant enough changes over time for us to use that in drug discovery. So we're still working on it, and we've not given up on it, but there needs to be much more of a consortium effort, and that's why we're trying to build teams around it to compare from one clinical center to another to be sure that we get standardization of these techniques and we're not missing something. Another approach, because we're not only worried about the brain, is the spinal cord imaging, and that hadn't actually been done at all because most of the diseases, MS and Alzheimer's, they'd focused on brain imaging. So Michael Benneter and a few others have been really the first, through our support, to develop imaging for the spinal cord. And it's still in the beginning stages, there's still a lot to be done, but both chemically and physically, there are clear changes in the spinal cord. Another area of interest, of course, is to see if you can do a blood test or a spinal fluid test and find a molecule or a signature that changes. And one thing that's very interesting is a particular component of that long axon called neurofilaments. And this definitely in a mouse model increases significantly with age. And we were able to show a similar change in human patients, where we have here our control. You see it's fairly low, and in ALS, although it's very variable, it's always significantly increased. So that's a fairly good marker. It's not so specific, because actually for traumatic brain injury, this kind of thing would be identified as well, this very high level of neurofilaments. So the specificity is not, it wouldn't be a diagnostic for ALS, because you'd have to rule out other things. But it could be something valuable to see how it progresses over time. That was our idea. The challenge is that it is always high, but within a single patient, it stays high. So it doesn't change. It's just to give you the sense that although the questions are right, the science doesn't always cooperate, and it's not going to be easy. So these are the kinds of things we're thinking about. Again, showing you here control subjects, very low baseline. All of them are higher, but generally flat. So over time, it doesn't change. It is interesting, though, that Kevin Boylan, it's actually his work, showed that where these levels were extremely high, they tended to be the faster progressors. So it does tell us something about progression, but this would have to be done in many, many more patients to be sure, because you could send a very incorrect message if you didn't get this right all the time. Other areas that have been very well done is to look for more accurate and less painful, actually, because EMG measures are actually quite painful. This is to measure the output of neurons in a much more pleasant way. It's called EIMG. It's electroimpedance myography, complicated terminologies. But really what it's doing is if the tissue is healthy and you make a measurement, you'll get this healthy output. If the tissue is not healthy and a measure of how many motor neurons are dying, you'll see that kind of output. So there's a very distinct change where there's muscle weakness, and very easy to measure. And in fact, this is being tried in the Biogen trial, the Empower study, because before it can really be used in clinical trials, one has to validate it. And so we're hopeful if Empower, that study, shows an effect, and we see a change also in the marker, we might have a new biomarker for clinical trials. And then very briefly and finally, before I open for questions, is to look at a few of the clinical trials. There are more trials in this, but I thought, this is just to show the many trial sites throughout the US. We continue to increase the sites because we want to encourage as much participation. There have been questions in the past, why is it with this technology today that trials cannot be done in one's local area? That is true for some of the trials. We are trying to do the ALS-FRS scoring by telephone. It is possible to get nurses out to the home. Some of the trials, like the stem cell transplant studies, have to be done. People have to live within that region where the centers are. For obvious reasons, the monitoring is much more intensive and the procedure is much more intensive. But this is encouraging to see, and we support financially the infrastructure of these trial sites to make sure that there is a good participation in trials. So Empower is a study at Biogen. Many of you are probably familiar with this. As I mentioned, the compound acts on the mitochondria, we think. It's not a clear, definite, but most of the studies have indicated that that's likely what's happening. So we think it's improving the energetics of the cell, the health of the cell. We should have, by the end of the year, they'll have completed the data. I believe their last message to us was that in the early part of next year, we should know the outcome of the study. Cytokinetics is going into the next phase, a phase 2, with a treatment that is a small molecule, so you take it as a pill once, twice, or 3 times a day, and that has an effect to increase muscle strength. It's not proven yet, but it's possible that not only strength is improved, but it might extend lifespan. They have to do a bigger trial with more people to prove that, but that's the sense. I mentioned the stem cell study, and I mentioned in particular the ISIS study with the SOD, because I think it's so promising for the new C9 mutation. I really think there's a real opportunity to develop a therapy in a much quicker time frame. And then an interesting thing about this trial which we're supporting, New Dexter is taken for pseudobulbar effect, so the laughing and crying, the unwanted laughing and crying. And the clinician who was treating people with this symptom felt that he was hearing from his patients that those patients on this treatment were able to swallow much better. And so we're doing a study in bulbar patients to see if indeed this is the case, because although it's not a curative or extension of lifespan, it's certainly enhancing quality of life. And with all of this, quality of life is in many ways as important as extending, because if you're going to extend a poor quality, that's not something that we aspire to. So I think that's a very exciting and promising trial. We are also supporting a diaphragm study. The diaphragm is approved by the FDA because it's a device. We just want to feel, and the clinicians were very eager to be sure that we knew who were the patients that would benefit from it most, so that if we say that this is standard, we encourage you to do it, there would be a better message of, is it really going to improve survival? Because there have been some claims that it does, and there's not sufficient data. And who is it most beneficial to? In most cases, there's a certain breathing capacity that you should have before you do it, and there also should be a certain muscle capacity to be able to get the stimulator. I'm saying diaphragm pacer, and perhaps some of you don't even know what it is. It's a stimulator. It actually, in a way, stimulates the muscle to cause the breathing to happen. So unlike with a heart pacer, it's the same kind of idea in a way, very simplistically put. So there are very, very encouraging trials, and as we build more of these compounds and these drug discovery programs, more clinical trials will come. Yes, some will fail, but certainly the more we have that we test, the likelihood is there'll be a few of them that will be successes, and perhaps combining these different treatments will really be a success. And then finally, if you haven't been participating and you are interested, every month with the exception of this month, we have a research webinar. A scientist or clinician will come and speak about their area of interest, and I will guide the questions and answer sessions, and they've been fabulous. We have over 100, sometimes 200 people calling in. It's at a time between 4 and 5 on a particular day, and if you can't manage to call in because you're busy at that time, we have them all posted on the website, summaries and lay summary, and the recorded presentation. It is an opportunity. It's impossible. It's fabulous to be here today to talk in person, but it's impossible to be everywhere with so much going on. And actually sometimes it's really nice to hear from the scientists doing the work, and they make a huge effort to make it understandable, and they're right there to answer any of the questions. So it's been a fabulous opportunity to help get more research to you, to let you know that there's a lot of action. I couldn't even begin to cover what's going on at the moment, and this is just the key areas. In this particular one, there's a discussion about a blood biomarker that's very promising, and how inflammation is linked to ALS. And this group is actually developing both a biomarker and a treatment approach, and we're very excited about funding them in this area. So I know it's a lot to talk about and digest, but I hope that it gives you some sense, the realities of the disease, because I couldn't stand here and tell you this is easy, we've got it solved, and in a few years you'll have all the answers. But also the huge hope. I would be in a group of scientists, no more than 20 in 1993, discussing our next projects. We now get thousands together, and in fact annually the next meeting is coming up in Chicago. There'll be 1,000 scientists all getting together to discuss progress. So there's a phenomenal commitment, and every one of those scientists, be they clinicians, be they young people that we fund, are really committed and dedicated to finding an answer and a treatment and ultimately a cure. So I think there's a lot to be hopeful for, and I hope that I've answered some of your questions and happy to take a lot more. Thank you.